in person. And my name is Donna Brown. I had the pleasure of running this class this year with the students that you see here. Biology 403. It's a UH Manoa senior. It's in place of their senior thesis. So besides the month they spent here collecting data and writing their report, they spent 2 weeks before that at home on Zoom. We met every day and they learned a whole bunch of different fish and corals and seaweed and other kinds of invertebrates like sea urchins. And then they also read a lot of papers that had background information about Kaneohe Bay and other studies that have been done to give them a background for this. And then they came here and were immersed in the field. And the first week we spent exploring, and then they came up with a project. So today they're going to present their project. And I introduce you the class of 2025 Biology 403.
Hello, and good morning, everyone. Just wanted to say thank you for Donna for introducing us, and also thank you all for taking the time out of your day, whether in Zoom or in person, to get here for our presentation. Like Donna said, it's the final day of our class, Field Problems in Marine Biology, and we're all so happy to be here. I had the lovely opportunity of working with my 9 colleagues for this 10-person team. But my name's Yasmin.
I'm Hannah.
I'm Sophia.
I'm Caitlin. I'm Isabelina. I'm Jenna.
I'm Brett.
I'm Molly.
I'm Orlando.
And I'm Hannah.
All right, to give you guys some context on Kaneohe Bay, it is a semi-enclosed bay where ocean water comes in as well as the fresh water from inland, and it mixes to create this really diverse marine habitats for all these organisms here. However, because of that unique placement of where the water mixes, it does make the reefs here a little bit more susceptible to human and anthropogenic factors. So, for example, there's been a lot of dredging here. There's been agriculture runoff as well as sewage runoff. And so all of this has factored in to change not only the reef's landscape, but also the chemical processes in which these organisms act in this ocean. And so if you combine all 3 of those human factors with climate change that is also happening, it has led to an increase in the nutrient influx, as well as the sedimentation, which covers the coral and can suffocate it. And also ocean acidification, which is just acidification of the water. And that has led to bleaching events here in Kaneohe Bay, as well as it's been seen that the reefs are turning from coral dominant to algae dominant. However, these reefs are incredibly resistant and resilient, and it's such an important place to have reef conservation strategies studied. And looking at this is beautiful Moku'olo'i that we are here today. It's been a marine protected area since 1953, and here you can see all of the reefs that surround the island.
So when we first got to this island, we spent the first week doing explorations. During those explorations, we made observations of the reefs that we were looking at. And based on those observations, we developed some questions. We questioned if the percent cover of coral and algae is higher in the patch reefs than on the marine protected area reefs around Moku'olo'i, and if there's a higher diversity of reef fish on the reef crest versus on the reef flat. We were also curious if benthic cover had any effect on fish abundance. So based on these questions, we developed some hypotheses. The hypotheses are that the percent cover of coral will be higher in the patch reefs than on the marine protected area reefs, and that the percent cover of algae will be higher on the marine protected area reefs than the patch reefs. We also hypothesized that there will be a greater fish diversity on the reef crest than on the reef flat, and that if there's a higher coral cover on a reef versus a lower algae cover, there will be a greater fish abundance. So there will be a greater fish abundance on the patch reefs than on the marine protected area reefs.
So to even begin answering these questions, we needed to have areas in Kaneohe Bay for us to study, compare, and analyze. In the northwest area of the bay, we chose to study 3 sites of patch reef locations, and then we chose 2 sites on Moku'olo'i in the southeast part of the bay, Gobi Bay and Coconut Island Beach. For some context, this is a general profile of what a reef looks like. There are four main parts of the reef slope, the drop off, the reef crest, and the top or the reef flat. The areas that are found most often populated with corals are the drop off, the reef crest, and the reef flat. And so the reef crest and flat are what we chose to focus our study on. These are the three patch reefs in the northwest part of the bay that we chose, patch reef 26, 27, and 28. We chose these locations because they have very close proximity to each other. They're very topographically similar, and they were used in the past for benthic surveys with the Department of Aquatic Resources, or DAR, for a biocontrol urchin experiment in which macroalgae coverage was being seen as whether or not it can be controlled with these urchins and manual removal. Patch Reef 28 was the control site, and Patch Reef 26 and 27 were the sites that were used in the experiment. So with this history of benthic survey, we thought that it would be an ideal site for us to follow up on in our project. The marine protected sites that we chose at Moku'oloi were Gobi Bay and Coconut Island Beach. Similar to the patch reefs, we had this sandy flat on top with the reef edges that were very accessible and easy for our class to study within our means. All sites were studied using 3 transects on the side edge of the reef or the reef crest, and 3 transects on the reef flat on top.
Well, for each of our 5 sites, we follow the same methodology involving the use of 6 10-meter transects. Once we arrived at our first area, we would lay out our initial 10-meter transect for our fish survey. This involved having 2 of our team members swim 2.5 meters on both sides of the transect and record individual accounts along with species on an underwater slate.
Some of the species that were observed during these fish surveys are shown here. We have the Indo-Pacific sargents, gold ring sturgeon, and also juvenile parrotfish. It's also important to note that during these fish surveys, there were very few adult parrotfish that were observed.
Once our fish surveys were concluded, our quadrant team would move in onto the same transect, and they would place and photograph 10 randomly placed quadrats. During photography, our fish team was moving on to the next area on the reef and laying down the next transect for the following survey.
And this kind of just gives you guys some perspective of what we were looking at while we were snorkeling. So for example, this just shows the reef crest and it's heading down into the slope. So that's where we would do half of our transects. This is the reef flat where it's nice and shallow and kind of just keeps going. We did 3 transects there. And this is some Parietes compressa coral within some 1 Montipore capitata colony growing. And then down here in the bottom right, this is kind of just an overview of what we would see a lot of the times. This is just several colonies of Parietes compressa.
All right, for our metadata, we recorded the times of the surveys, which included start time and end time for each transect. We also recorded GPS coordinates at the end of each transect line and weather conditions and tide height. We were able to use a dive computer or a dive watch in order to get water temperature at each site and the maximum depth and the minimum depth of each transect line. And with our GPS coordinates, that's the images that you saw earlier, the Google map of each of our sites. Those little blue dots are the GPS coordinates of each transect line. And we also gave the quadrat team their 5 randomized points for each quadrat that they set down on the transect line.
In marine biology and for our project, photogrammetry is a very useful tool. It helps to permanently capture the study site. So in this picture, we laid down transects to measure out a 5 by 5 meter area. At each corner, we laid down GCPs. GCPs are ground control points, and they let this photogrammetry software to be able to size and scale the models that we create. And at each GCP, we collected the depth and the coordinates. And then in this picture, you can see me holding a GoPro up straight flat down to capture the reef. And I went in a mow the lawn like motion to capture a time-lapse video of our 5 by 5 meter area.
Here's just a quick overview of our methods, starting with the top left of our 10-meter transect line, conducting our fish surveys. Down here, we have our 0.5-meter quadrats, taking pictures, and then over here, we have our 5x5-meter area with our photogrammetry structure of motion with the 4 GCPs as Jenna went in a mow the lawn motion up and down.
For the benthic cover, we use the CoralNet program, which is an annotation tool utilized for analyzing benthic imagery. The system has a randomly generated point system, and for each image, we did 50 points that were then categorized to be put into the system to calculate the total percent coverage of both coral and algae at each site. The program has its advantage of being a standardized analyst process of being consistent and repeatable for each image. These benthic surveys were then used as a baseline for our other components in our study, such as fish surveys. Moving on to results, we start with our benthic cover analysis. On the first graph here, we have the mean percent coverage of both coral and algae in each of the 5 surveyed sites: Coconut Island Beach, Gobi Bay, Patch Reef 26, 27, and 28. In the bars, we see the coral coverage shown in blue and the algae coverage shown in green. While there is coral dominance in each of these sites, the algae is still a large proportion of our benthic community. Moving on to the next graph, we switch from individual comparison to reef type comparison with the marine protected areas and the patch reef areas. This is also comparing the mean percent coverage of coral and algae. And similar to the individual site breakdown, we see a pattern of coral cover being larger than the algae cover, showing that even though the marine protected areas and patch reefs are different, that the marine protection areas have the protection status showing higher coral dominance.
This graph demonstrates the 5 most common species of coral that we found during our analysis. Porites compressa was the most abundant coral by far, representing over 80% of our corals in the spot. Altiplora capitata was the second.
Sorry, wait a second. I don't think I can hear it. Start with the first.
Yes. Porites compressa was the most abundant coral that we observed during our study, representing over 80% of all corals observed. Montifera capitata was the second most abundant coral species.
I don't know where to put it.
And Poecilophora democordis, Latastria porphyria, and Lovactis scuteria, while we did observe them anecdotally swimming around collecting our data, they were not very.
I'm sorry. Oh.
They were not contributing greatly to our data because they were not seen as much during the quadrat data. They were only contributed about less than 1% to all of the coral cover that we found.
This is the abundance for individual algae species across all 5 sites. A couple notable things of this graph is that turf algae and crustose coralline algae were the most dominant across all 5 sites. And then another species of note is the Capophycus and the Euchemia. These were the 2 that were manually removed during the DAR project on Reef 27 and Reef 26. So it's interesting that they were only spotted on Reef 27 and Reef 26. Also, when comparing the raw data that we got from DAR, immediately after their project, it said that the combined percentage of Euchemia and Capophycus was around 1% on Reef 27. But now, as you can see, it's around 8% combined for Reef 27. So there is a subtle increase there. As well, another invasive species, the Gracilaria salicornia, that one, immediately after, before the treatment, it was at 3% for Reef 27. And then after treatment, it was at 7%. Now, it's just about 1%. So that is very good, especially since we didn't see many urchins on the reef, but it seems like they are working. Another species of note that was brought to my attention is the Dictosphaeria versluzi. That's a native species of algae, and we saw it across all 5 reefs. So that is very good that we're seeing that, and hopefully it continues to flourish.
So fish diversity is mainly controlled by 2 factors, abundance and richness. Abundance is the total number of individuals counted, whereas richness is the total number of species recorded. So going off that, we notice that our patch reef sites contain the highest abundance and richness, so we conclude that the highest diversity was also present on our patch reef sites.
Going into this, we see the mean fish abundance comparing to both reef types of marine protected areas and the patch reefs. What's seen here is the average with the black dot for both reef types. And noticeably, there is a large variability in fish abundance in the patch reefs, which means that during the transects, some fish were reported, many fish were reported, and some were not reported at all. What's shown in the marine protected areas is a narrower fish abundance, meaning it had more stable condition fish populations. Moving on to mean fish species richness, the marine protected areas is slightly larger than the patch reefs by about 1 species on average, but with the error bar overlap, this shows that there is not a statistical significant difference. With the reef types of marine protected areas, there is the protection status showing that it can help maintain higher fish diversity.
So we organize our fish data into their respective families here on the x-axis with their total counts on the y-axis. And here we can see that the patch reefs do show high diversity overall, with an exception being the Gobiidae family, or gobies, which showed higher abundance on the MPAs. This is likely due to the extensive sand flats that are present on the MPA sites here on Mokololoi, such as Gobi Bay, where gobies tend to make their burrows. This is again organized family on the x-axis with counts on the y, but this time looking at reef flats and reef crests. Here we see that there are more carnivorous and omnivorous fish, there are more herbivorous fish, such as our damselfish, parrotfish, and our tangs and sturgeons on the reef crests. We also note that our We also know that there are more carnivorous and omnivorous fish, such as our snappers, our damselfish, and our wrasse were more prevalent on our reef crests. Here we separate our fish counts into their respective trophic groups on the y-axis with our counts on the x. And we can see that our herbivorous fish were the most dominant across the board on both reef flats and reef crests. More carnivores are present on the reef flat. This is likely due to a high abundance of Asteroteryx semipunctata, which is a half-spotted goby. We note that there was higher omnivorous presence and corallivores on the reef crests, where there's higher abundance of live and more diverse corals as well.
Coming back to photogrammetry, we used Patch Reef 28 to capture a visual representation of the reef sites we were looking at. So this is a 3D model created in a software called Agisoft Metashape Professional. And in this 3D model, it really captures the 5x5 area that we surveyed and took data for. And you can see the GCPs in each corner of our 5x5 area.
Play it again.
You can also see a good visual representation of that reef flat compared to the reef crest on the side. From the same software, an orthomosaic model was also created. This may look like a normal picture taken, but it's actually the time-lapse video from the GoPro and around 1,200 photos with almost 2 million tie points to create this image. In the same area as the orthomosaic from the last slide, this DEM or depth elevation model was also created. It's a representation of the shallow -0.2 meter to the deeper -3.3 meter depth profile.
I'd like to now move into the discussion where we'll explore what these findings might mean. From our fish surveys, we found that herbivorous fish were the most abundant trophic level across all of our reef sites. On the patch reefs, herbivorous fish had the greatest diversity, whereas carnivorous fish were more prevalent at the Mokolo'e sites. As we surveyed the reef crest and the reef flats, we were able to determine that the reef crest had a higher fish diversity. Whereas the most abundant species that we saw on flats were initial phase and juvenile parrotfish, which can suggest that these flats serve as a nursery for these young fish. Based on our findings, all of our sites except for Patch Reef 27 had a high coral cover dominated by Parietes compressa. However, on Patch Reef 27, it expressed a even coral to algal cover. And one observation that we made was the lack of collector urchins. As previously mentioned, in 2011 to 2013, DAR undertook a project that involved outplanting almost 100,000 collector urchins on Patch Reef 26 and 27 as a biocontrol effort on Gracilaria salicornia and Acanthopteris spicifera. These two algae species are highly invasive across Oahu, especially on these patch reefs. However, across the entire 75 square meters that we surveyed, we recorded 0 collector urchins and observed no more than 2 individuals while surveying our sites. As seen in our results, Patch Reef 27 had an unexpected high algae abundance compared to the relatively low algae abundance on Patch Reef 26. It should be noted that the majority of the abundance consisted of turf algae and crustose coralline algae. However, some invasive species like the Acanthopteris spicifera reached almost 5%, which is similar to the percentages found previously before the DAR biocontrol project. Some possible reasons for this unexpected high algal abundance on 27 could be due to the lower abundance of herbivorous fish compared to Patch Reef 26. Herbivorous fish are important as their diet consists of algae, which can help maintain the cover across the reef. Also, the lack of collector urchins present on the reef could contribute to this abundance as they were specifically outplanted to attack invasive species like Acanthoptera. Thus, with the lack of collector urchins, Acanthoptera has the ability to then proliferate.
So some future directions that we can take is using non-visual methods such as DNA metabarcoding and acoustic surveying. Most of the times that we took our surveys were in the morning or early afternoon, so we did not account for any cryptic or nocturnal species. Using these non-visual methods will help us better understand what life is like on these reefs.
And further future directions is doing repeated surveys to capture variability, like Isabella Elena just mentioned, doing different times of day, like earlier morning or even night, doing it over different seasons or over a longer period of time, like years. Also continuing to collect detailed environmental metadata, like temperature, light, and nutrients can provide valuable insight into reef health and what impacts reef health. Also integrating socio-ecological data into your surveys like fishing pressure, coastal development, and community practices also gives insight into anthropogenic factors and how that can also affect reef health. Another important future direction is including more sites in the frame of reference during surveys. We surveyed 3 out of the 54 patch reefs in Kaneohe Bay. So including more patch reefs and more marine protected areas in multiple reef zones can expand your data set and lead to successful surveys.
Now let's move into our conclusions to see how our findings fit into our original questions. Revisiting our hypotheses from the start of our study, for our first and main hypotheses, We decided to reject the null because there was no significant difference in the coral and algal cover between the MPAs and the patch reefs. However, for our second hypothesis, this was supported by our results as there was a greater fish diversity on the reef crest. There was a higher greater fish diversity in the reef crest compared to the reef front. For our third hypothesis, we decided to also reject this null because there was no correlation between coral and algal cover with fish abundance. Additionally, there was no significant difference in fish abundance between the hatcheries at Kaneohe Bay and the MPAs at Mokuoloi.
Our surveys are important in helping maintain the health of coral reefs. Coral reefs are extremely vital to the Hawaiian Islands as they serve as a protection to its environment and its communities. The Hawaiian Islands experience strong wave forces that can cause coastal erosion, flooding, and damage. And these reefs act as barriers that dissipate these forces to help mitigate these damages. Therefore, our surveys are important as they serve as a baseline for future management practices. Conservation efforts can use our surveys as monitors to monitor changes in coral reefs, coral cover, and spread of invasive algae.
Special thanks to our two instructors, Donna and Dr. Hanner, and our team, Madeline, for their continued support and guidance throughout this project. And thank you to HIME faculty and staff and HIME instructors for their continued help, as well as teaching us new valuable skills throughout this course. And special thanks to HIME as well for providing all the necessary resources and facilities required to make all of this possible.
And we just wanted to showcase some of our photography skills that we learned throughout this course. That was honestly one of the most fun things about taking this class. So we just wanted to, highlight some of them. And so we have a Christmas tree worm done by Sophia over there. There is a school of Hawaiian bacillus by Jenna. And then next up is a snowflake eel, nice close up by Brett. And last is a feather duster worm with a sailfin tang in the back by Hannah Walk over here. So we just wanted to thank you all so very much for coming. A lot happened to be able to make this all, work. And we're just so grateful for the experience of this class. But any questions, comments, or favorite parts? Yes.
Yes, sir.
Yes.
Beautiful talk, thank you. I was wondering, based on what you saw, would you recommend collector urchins as a strategy for controlling algae?
Wait, what was that?
Yes, super.
If you didn't hear on the Zoom, he asked if we would recommend collector urchins as a future, biocontrol, do it again. Based on the cost that we saw that DAR spent, I believe it was around $800,000 for their initial urchin outplant. So I would say that there could be better means. I do believe that the increase in the biocontrol and increase in that is a hard word, herbivores, will help with invasive algae. So I do believe that that can be a study that can be revisited and more outplants can happen. But I think the cost would have to be supplemented somehow, because I think the reason it hasn't been repeated since is because of the cost, and that the results, I think, were not as significant as they wanted them to be. So yes, I do believe that it is a good idea. And we wish that we saw more urchins as well.
Yes.
Great job. It's kind of a follow-up question. I was just wondering what's happening now with the urchin hatchery and where all these urchins are going.
I tried to get in contact with the Department of Agricultural Resources and talk to see if this was still ongoing. I did not receive any response by email or by phone call about the project, unfortunately. So what we know is that this was last, like the information about this was last revisited around 2017. But the project of the urchins went from 2011 to 2013. So it's definitely been a while and could use some revisiting for that project.
Yes.
So your guys' first hypothesis was about the coral cover between the NPA site and the non-NPA site. Why do you think that there was not a difference in coral cover? Like, why should NPA status matter? I guess is a two-part question.
I think maybe the answer that would like formulate with that, because it was kind of shocking that there was no difference. Like that was kind of, we were kind of like, oh shoot, our data is not significant. We were like, oh no. However, the patch reefs did have a steadily higher algae cover. So despite the percent of coral cover not being too different between the two, the algae cover was still higher on the patch reefs. So I think that does show a little bit that marine protected areas do need to remain marine protected. And even though this is a marine protected area, that's not enough. This needs to be a marine protected bay. This needs to be a marine protected island. Like there's still stuff coming into the bay that's affecting this island, even though it's protected from outtake. So I hope that answers your question.
Yeah, I guess like the NPA restrictions though are no take and no fishing, right? So have you guys looked at what fish or like what food fish are more abundant on each site? Because that might give you some more information if like NPA is actually working. And maybe Brian can talk more about that. I think that the more like what are people fishing would be helpful to know as well.
To follow through on what Professor Barr said, we have done what you identified as the future. We've done the first round of environmental DNA metabarcodes on a protected area in Waikiki. And we found no difference inside and outside the marine protected area. And our conclusion is not that the marine protected area didn't work. It's that it's too small. Given the size of these ecosystems, that protected area is just a few acres. It's a postage stamp. And so that kind of data is not going to tell, I don't think it should be interpreted as whether the protected area works or doesn't work. It's how big do they have to be to work.
Thank you for that insight. That's great questions. That's stuff we even think about. Awesome. Well, is there anything on the Zoom? But
We don't have anything on the Zoom.
I have a question for you. Just, I know you weren't surveying turtles, but like, how many turtles, did you see very many turtles out on the patch reefs?
yes.
Yes, we did.
Yeah.
I think
And
Jenna
they
counted.
especially like to eat grass.
For the turtles that we saw on these reefs, it was specifically on the patch reefs 42, 43, and 44. And specifically on 43, I counted 12 just on that reef alone. On 42 next to it, I think there were around 3. And then for the patch reefs that we surveyed, 26, 27, and 28, I'm pretty sure the last reef that we surveyed, which was 27,
Yes.
right? I think we saw maybe 3 turtles, so not as many over there. And then in retrospect, where our patch reefs were, 43 were further out, further north.
I would also like to add on to that at patch reef 44 and 42, we observed a lot less algae than we did at our sites and here at Mogul.
To follow up on Don's question, you guys notice any like weird behavior or what they were doing around those patch reefs?
Most of them were in the burrows, like in their little coral nooks that they like to make, like their little holes. If it wasn't that, it was mostly them coming up to the surface. But like, oh yes, tumors.
We
on,
saw
like their
tumors on a lot of
faces. We did see one without a fin. That was heartbreaking. But yeah, nothing too out of the ordinary. That was your guys' project last year, right? Yeah. That's funny.
A lot of barnacles.
But yeah, thank you guys.
Ask if anybody on Zoom has a question.
Anyone on Zoom have any questions? I don't see anything in the chat. No, okay.
Thanks, everyone.
That was so good. Congrats, guys.
Where's the champagne?
No, like for spring.
Oh, people on Zoom.
I knew it. You look just like her.
Yeah.
Talk to your mommy.
Do you have a question? Oh, okay, let's stop this recording.